Hi, welcome back. So, for the last segment of the tutorial, we're going to talk about viscoelastic materials and cells. This is part of the tutorial on fluids. Now, as you recall, we already spoke about Newtonian fluids and Newton's law of viscosity. So, what in some senses are viscoelastic materials? And we mentioned very briefly that they're materials that show both solid-like and liquid-like properties. Now, Oobleck is something that you can assemble in the kitchen. It's a viscoelastic material in the formal sense. It consists of a 2:1 ratio, volume by volume, of cornstarch and water. And to do that, to prepare it, you need a container to mix it. So if you're in a laboratory, a beaker will be fine, and a petri dish, a large petri dish into which to pour it and demonstrate some of the properties. We will talk about this when this class goes live. You can play around with this quite a bit and you'll find a lot of popular YouTube videos on this. For example, putting this Oobleck on a plastic sheet on top of a loudspeaker with noise at increasing frequency generates very unusual patterns. The part that we're interested in is that when we perform slow movement, like we take a spoon and move it through the Oobleck, it behaves like a liquid. If we now, however, try to cut it very rapidly with a high amount of force and with high acceleration, you will find that the Oobleck, this cornstarch water mixture, behaves more like a solid. And this is broadly called shear thickening. That is to say, the shear force aligns the molecules that make up the material, creating almost a solid-like geometry. When we remove the force or reduce it, then it returns to a more liquid-like behavior, which is disorganized and in motion. And this can be cyclically done. You can go back and forth between solid-like and liquid-like. So you could argue that the nature of deformation determines what behavior you will see. A counterpart to shear thickening is shear thinning, where the effective force is the opposite. Instead of making it more solid-like, it makes it more liquid-like. And instead of removed force making it behave more liquid-like, it behaves more solid-like. In that sense, the behavior of simple kitchen-made reagents like Oobleck may be a bit simple. So what about cells? Now, one of the many tissues in the human body that need to withstand stress and strain and mechanical deformation are chondrocytes. They form cartilage and are part of the mechanical tissue in vertebrates that respond to stress and strain. Another very obvious example is muscle cells, heart muscle cells, skeletal muscle cells. So here you're looking at a picture of immortalized mouse artificial chondrocytes, IMAX, and experiments that were done on chondrocytes in culture that demonstrate that their behavior is viscoelastic. So the experiment initially involved isolating the nucleus of a cell. So what you're looking at in A is a whole cell, and by successive A, B, C, aspiration and expiration and aspiration and expiration, the workers Oobleck and company could destroy the cell body. In fact, to such a point that, as you can see in D, they got a nice isolated nucleus. The approach that they took was also compared to chemical isolation, and they found very similar results in terms of morphology. How do you check that this nucleus, however, is a bona fide nucleus? Obviously, you want to look whether the molecules are the correct ones inside it. The molecules inside the nucleus are nothing but DNA. So one straightforward way the Oobleck and company team tested the validity of their method was to stain these nuclei with a DNA dye, acridine orange. Today you would use Duppy or Hexed. And they saw that the nuclei are modified. So they went to the next step, which is to modify, deform the nuclei and compare the deformation to cells, whole cells. What you're looking at here is a single nucleus caught in a small 2 and 3 micron bore inner diameter micropipette tip, and with a constant pressure difference applied across the micropipette. What happens, as we can see, is that rapidly the section of the nucleus deforms where the arrow shows in B at 2.54 seconds, 2.54 minutes probably it is, and at 2.59 minutes, 2 hours 59 minutes, so in about 60 seconds, the deformation reaches a maximum. And this is what they plotted in the graph below, where the aspiration length, that is the length that enters the tube, increases initially linearly and saturates. This is true of both the nucleus and the cell. However, for the nucleus, this is a far smaller range and a faster kinetic as compared to the cell. This initial linear deformation and slower timescale of relaxation is characteristic of viscoelastic materials, and the team of Oobleck and company fit a model of 2 elastic constants and 1 drag coefficient to estimate the modulus of elasticity of the material and the drag viscosity, effectively the viscosity of the nucleus. They found that this was higher than the whole cell, and it was independent of whether the nuclei were chemically isolated or mechanically. So with that, we end our sort of brief introduction to viscoelasticity, and we will return to it at another time. With this, we end the tutorials and return to theory. As we discussed, the use of cornstarch water mixtures can be used to demonstrate Oobleck and non-Newtonian viscoelasticity. So in this following video from MIT Boston laboratories, you will see some interesting theories that then relate to a research paper published in their lab.
You mix cornstarch and water, weird things happen. Swish it gently in a bowl, and the mixture moves like a liquid. Squeeze it, and it starts to feel like paste. Roll it between your hands, and it solidifies into a rubbery ball, until you try to hold that ball in the palm of your hand, and it loses its structure and dribbles.
And this is what we refer to as shear thickening. These demonstrations are things you can do in your kitchen experiment.
Dribbles away. To many who have played with this material, perhaps as children, its strange behavior is nothing new. But understanding exactly how, why, and when this material will act a certain way has always been rather unpredictable. But now, a team of MIT engineers have developed a mathematical model that can accurately predict this material's behavior under various conditions. A single particle of
So as you know, mathematical modeling of physical systems is a very common way to engineer them. And this is really the crux of quantitative biology, to mathematically model it using physical theory so that we can make predictions and even make modifications in a way that we understand them. Now, the theory that is behind this is a little more complex than we have time to go into, but I recommend you to read the paper in case you're interested. This is not part of your regular coursework as such, but this is extra reading.
Cornstarch is about 1 to 10 microns wide and about 100 times smaller than a grain of sand. It turns out that particles at such a small scale experience effects that larger particles do not. Because cornstarch particles are so small, they can be influenced by temperature and by electric charges that build up between them, which causes them to slightly repel against each other. So as long as you move slowly, the grains will repel and slide past each other like a fluid. But if you do anything too fast, you'll overcome that repulsion, the particles will touch, there will be friction, and it will act as a solid. The researchers incorporated equations into their model to describe the effects of particle repulsion and the speed at which the material is deformed, to predict whether it would behave as a solid or a liquid under various scenarios. The researchers say the new model can be used to explore how various ultrafine particle solutions, such as cornstarch and water, behave when put to use, as for instance, fillings for potholes or bulletproof vests.
The paper relating to this study can be found in the classroom environment, and it is from a journal, Proceedings of the National Academy of Sciences, USA. The year of publication is 2019. The authors are Baumgarten and Cameron. The equations, as I mentioned, are a little complex and they go beyond the scope of this course, but you're welcome to read it.